It's a great honor for me to introduce Professor Elizabeth Buffalo. She's a professor and the chair at this moment in physiology and biophysics at the University of Washington, a very famous department, great department. And she's the associate director for the Primate Center at UW. She went to Wesley as an undergraduate, born, as you heard from our conversation, in Arkansas, and went to the famous Central High School. And if you don't know anything about Central High School, use your Wikipedia and look it up. It's a site of integration in this country for high schools. She went off from UC San Diego, where she earned her PhD, then to NIH, and she's really one of the leading investigators in the world in cognitive neuroscience, and she's made some very interesting observations and contributions to memory. And of course, being in a department with the Ojemans, it's a great overlap of work. She's extremely well funded from NIH, the Simons Foundation, McKnight Foundation, Pfizer. She's just a great partner and a great colleague to have in the field, and her overlap between what we do and what she does is pretty fascinating. So I'm not going to take any more of our time other than to say in 2011, she won the Trollard Research Award from the National Academy of Sciences for her innovative work. So Beth, thank you very much for being here. And you have the screen, beautiful picture. Thanks for Thank you again for getting up so early too.
Of course, thank you for that, that really kind introduction, and thanks for inviting me to speak for you all today. It's interesting when I think about my first visit to UW when I was considering moving from Emory. I met with George Ojeman and got to hear about, he's an icon and a real, I was a real fan of his work, and so I was really excited to get to meet with him. And it's been amazing to be able to collaborate with Jeff. We're on a project and co-mentoring a student, so that's been really fun. So today I'm going to tell you about some of the recent work in my lab. I'm going to tell you a little bit about some published work, and then also show some unpublished work and kind of tell you where we're going and where we hope to be in the next couple of years. And the focus is going to be the hippocampus, so that's the structure that we study in my lab, and trying to bridge the gap, as I say, between these two views of the hippocampus, the spatial view and the mnemonic view. So just to kind of introduce these, I probably, to this group, don't need to introduce patient HM. He is considered the most famous amnesic patient. He received a resection of his medial temporal lobes bilaterally in order to relieve otherwise intractable epilepsy. And so, and what was discovered after this operation was that he suffered from severe memory impairment. And this case really led to really launched the modern era of the neuropsychology of memory, and firmly established the idea that the hippocampus and other medial temporal lobe structures are really critical for certain forms of memory, particularly declarative memory. But at the same time, work that was begun in the early 70s by John O'Keefe and colleagues really focused on a different aspect of hippocampal function. And this was work that was primarily carried out in rodents using single unit neurophysiology. And in this work, they really focused on the spatial representation of this area. So recording from single units, they identified beautiful spatial coding in the hippocampus and the surrounding cortex. And this has led to the idea that the hippocampus is really important for navigation and for forming kind of a spatial representation of the world. And so in my lab, we're working in the non-human primate, mostly the rhesus monkey, and looking at these same structures in the medial temporal lobe, and trying to sort of bridge between these two ideas. So the spatial view has predominantly focused on work in rodents and single unit neurophysiology, and the mnemonic view has primarily focused on studies from patients and human patients. And so we see that our work in the monkey, where we can train monkeys to perform some of the same cognitive tasks that can be performed in humans, while also using single unit recording techniques, may serve to kind of bridge between these two ideas. I won't have time to talk about it today, but some of the exciting work that we're doing with Jeff now, Jeff Ogeman, involves single unit recordings in the medial temporal lobe in human patients. And this is going to be a further way to help us sort of understand these ideas and bridge the gap between these two views of the hippocampus. So just to give you an outline of the work I'm going to present for you today, in the first part, I want to tell you a little bit about rhythmic neuronal activity in the hippocampus and what we've discovered in the primate hippocampus, how this activity is associated with a certain form of exploratory behavior. And then in the second part, I'm going to talk about the spatial representations that we've identified in the primate medial temporal lobe. And then the third part, I'm going to talk about our ongoing work, where we're moving into using new tools and techniques, including virtual reality, to better probe what's been called the cognitive map. And I'll define that a little bit as we go along. So our work uses eye movements to index cognition in monkeys. And just to give you a sense of our motivation for this, a lot of this goes back to the work of Alfred Yarbus from the 60s, where he started measuring humans' eye movements as they looked at different images, in this case, a painting. And he discovered that the eye movements and the precise pattern of exploration of a scene could tell you a lot about internal cognition. So in this example, he asked the subject to just examine the painting freely. And the white trace here represents the subject's scan path as they're looking at the image. And then in a second showing of this picture, he just asked the subject to estimate the material circumstances of the family. And here you can see that a completely different pattern emerged, where the scan path was looking at different parts of the scene. And so we decided to use this tool in our recordings with monkeys to look at memory. So there are several studies that suggested that the way that you look at an image the second time you see it compared to the first is quite different. And so we could use this technique to index memory in monkeys. So in our experiments, we have the monkeys seated, their head fixed, and they're looking at a computer monitor. And we're measuring their eye movements as they're viewing these images while we're recording the neural activity from deep in the brain, from medial temporal lobe structures. And here's an example of the kind of behavior that we get when we look at the scan pattern and the viewing behavior for novel images versus repeated images. So in this example, the yellow trace shows the monkey's scan path the first time that he saw this picture. So you can see he looked at quite a few of the features. And the blue trace shows the monkey's scan path the second time. So here the monkey just made a few fixations, made a few eye movements, and then looked away. And this is what we found across many monkeys, thousands of images. We see this preference for viewing novel images compared to repeated. And this is an innate preference that, that many species have. Monkeys, humans, even rodents have a version of this that I know David Bass, who's on the call, worked on when he was a graduate student. And so this gives us an index of memory that we know depends on the hippocampus. And so we can look at activity in the hippocampus while monkeys are performing this task to get a sense of what signals might underlie this memory performance. Now, when rats are performing as a kind of exploration task, there's a prominent signal that's been identified in the hippocampus, which is the theta band signal, or the theta band oscillation. And an example of this is shown here. So this is this low frequency oscillation that you can see, or you can hear in the hippocampus when you're recording as the rats are moving around and exploring an environment. And then this signal goes away, you get a different kind of signal in the hippocampus when the rats are still, when they're engaged in grooming or eating or some other kind of behavior. Well, in monkeys, we don't really see this kind of theta band oscillation, this prominent oscillation. And if you think about it, when we're recording from the monkeys, they're seated, they're immobile, they're not moving around. And so the idea has been, well, we don't see this prominent theta band oscillation because the conditions that we have are just the wrong ones for eliciting that kind of oscillation. But in our experiment, even though the monkeys were immobile, we allowed them to move their eyes. And so here's just an example of two scan patterns as they're viewing images. And we wondered whether, we had the idea that maybe as they're exploring these scenes, we may have a chance of seeing something like a theta band oscillation because they're engaged in the same kind of cognitive task, exploring their environment. And when we started looking at the details of how they moved their eyes when they were exploring their scenes, it was interesting to us that monkeys and humans tend to move our eyes in a sort of pseudo-rhythmic way. And so this is showing a histogram across many images that the monkey's viewing. This is looking at the duration of fixation, where the median is right about 200 milliseconds. And you can see it's this sort of pseudo-rhythmic behavior where the monkeys are moving their eyes at about 4 to 5 hertz. And this is really right at the lower end of this theta band oscillation. So we wondered if there might be something in the signal that we could record from the hippocampus that might be related to this eye movement behavior. And so we just looked at what's called the local field potentials. This is kind of the local EEG in the hippocampus as monkeys were doing this free viewing task. And here I'm showing you individual trials which represent individual eye movements. So here the dotted line is the time of the eye movement. The red is the raw trace, so the raw local field potential. And the blue is just this trace filtered in the theta band, the low frequency band. And what we found was that prior to a saccade, this is the saccade again, the dotted line, there was unlikely to be any relationship in the phase of this oscillatory signal. But after the saccade, there appeared to be a phase reset. So now across individual eye movements, there was more of an alignment of this theta band oscillation. And so if we just average this, you can see that you get this greater theta band oscillation after the time of the saccade, indicating that phase reset or the alignment of the phases. And this is just the quantification of this. So on average, we get a greater increase in theta power after the saccade. And then we wondered whether this change in phase alignment might have anything to do with memory. Because again, we're recording these signals in the context of a memory task. And one thing that's interesting about this task that we're using is that there's no right or wrong answer. So the monkeys are just free viewing these images, and we're getting a sense of their memory based on how long they're spending looking at these images. And so we can just compare the change in looking time for the novel versus the repeated image. And the pictures over here, so this is the two different colored bars are two different monkeys performing this task. The images over here represent images that the monkey remembered well, so showed a big change in looking time for the repeated compared to the novel. Whereas these images over here are ones that the monkey didn't remember as well, so looked about an equal amount of time at the novel and repeated image. And so we could compare this phase reset well-remembered images compared to the ones that were not well-remembered. And what we found, this is looking at the reliability of that phase reset. So warmer colors represents greater reliability. Time 0 is the time that the eye movement started. And at the top, you can see the pictures that the monkey remembered well compared to the ones at the bottom are the pictures that the monkey didn't remember well. And what we found was that there was a phase reset after the eye movement, but it was much stronger for the pictures that the monkey remembered well. So together, these data suggest that as the monkeys are moving their eyes and exploring the scene, every time their eye lands on a new part of the scene, you get a phase reset in this oscillatory signal in the hippocampus. And the reliability of that phase reset is predictive of successful memory encoding. So the idea is that maybe this phase reset goes to some optimal phase for the network to process the incoming information and form a new memory of that experience. So this idea that that this behavior of actively sampling the world might relate to the network activity in the hippocampus is not new. It's not something that we just discovered in monkeys, but had been known for many decades from earlier work in rodents. Rodents tend to sample the world by sniffing and whisking. And there were several studies, starting with work by Howard Eichenbaum and Macready's back in the early 80s, showing that there's a relationship between this active sampling of the world through sniffing and the activity in the hippocampus. And then more recent work from Matt Diamond's lab, this was published in 2015, showed that there was a relationship between whisking behavior when the rats were engaged in a discrimination task and oscillatory, this low theta frequency activity in the hippocampus. So there's this linking between the active sampling of the world and this network activity in the hippocampus. I mentioned to you earlier that we move our eyes at about the same rate that monkeys do. This is just showing this. This is data from humans performing different kinds of free viewing tasks, and showing that we also showed this kind of peak around 200 milliseconds for our fixation duration. So we move our eyes at about 4 to 5 hertz. And there's this interesting case report that was published back in the late 90s. And this was a case report of a college student, a young woman who was born with a degenerative disease of her eye movement, of her eye muscles. So she was unable to move her eyes from birth, but her vision was fine. And she made it through school. She was a college student at this time. And it turned out that she had developed a strategy of bobbing her head to move the image on her retina. And when they measured the duration of her intervals between head bobs, it turned out that it also fit exactly this profile. So she showed that her fixation duration, or her times of having her head still, were about 200 milliseconds. So this is when I start to sound sort of evangelical about theta oscillations, but you really find them in a lot of species and a lot of behaviors, that this low frequency 4 to 5 hertz rhythm, there's some sort of might be an optimal rhythm for receiving or processing incoming information. And work that we're still doing in the lab now is trying to understand more about how this behavior of actively sampling the world relates to that kind of optimal oscillation that we see in the hippocampus. So that's the end of the first part, where I wanted to focus on rhythmic neuronal activity and this low frequency activity in the primate hippocampus. But now I want to move on to spatial representations. So as I mentioned at the beginning, there are these two dominant views of the hippocampus, the mnemonic view and the spatial view. And in order to start to bridge the gap, we need to understand whether monkeys have similar kinds of spatial representations as those that have been described in rodents. And so just in case people are not familiar with these signals, I'll show you a movie. Let's see if the movie will work here. So this is John O'Keefe, who I mentioned, and his work back in the early 70s really launched the study of spatial representations in the hippocampus. Let's see if this works. So hopefully you can see that. So this is a rat just moving around a circular enclosure. And that popping sound you hear is the sound of an individual hippocampal neuron that's firing. And so as you notice, the neuron is very quiet, except when the rat moves into this location in the environment. And then it fires like crazy, and then it goes quiet again as soon as he moves out of this area, which would be called the firing field for this particular neuron. And the way we can represent this is also showing, this is in a square arena. The black lines represent the rat's trajectory through the environment, and then they would plot green dots wherever this neuron fired. Whenever the rat was in this location, the neuron was firing. And this is a heat map of this same activity. And so if you record from a bunch of these hippocampal neurons, then the idea is that you identify spatial representations that really map the entire environment. So across the population, you're recording all that activity. You could get a really good sense of where the rat was at any given time. And then in the early 2000s, Edvard and May-Britt Moser decided to try to address the question of where do these place responses come from? And so they decided to record from an area just upstream from the hippocampus, the entorhinal cortex. And here they found something very different. So in this video, you'll see the rat moving around a square environment. Here, instead of listening to the sound of the neuron, they've plotted a diamond on the arena whenever this entorhinal neuron fired an action potential. And you can see their student, this is Dory Durdekman, I think, throwing chocolate sprinkles into the arena to keep the rat going. And now you can see with this speeding up the movie a little bit, you get more of a sense of the responses. What you can see is that these are very different from hippocampal responses. So you see multiple places that this neuron likes to fire. And after about 40 to 50 minutes of recording, this beautiful pattern emerges of this firing activity. So you can see this really regular kind of grid-like activity of this neuron. So that's how they got the name grid cells. Here's another representation of this. You can see the heat map, the darker colors represent higher firing rate. And what they found was that these firing fields perfectly tiled an environment so that you could draw equilateral triangles between each of these fields. And it led support for the idea that this part of the brain, medial temporal lobe, the hippocampus and entorhinal cortex, really were important for navigation, for helping the rat move around and know where he was in the environment. But having just a sense of place or this kind of metric of the grid cell activity wouldn't really be enough to tell you exactly where you were. And so there were other cells that were also identified that support the navigation hypothesis. So a border cell, so these are cells in the entorhinal cortex that fire whenever the rat is moving along the edge of an environment, as well as head direction cells. So these cells really give the rat a sense of, or they fire selectively when the rat is heading in a particular direction. And more recently, velocity tune cells have also been identified. And so having all of these signals together really provide all the elements you need for the rat to use these signals to perform navigation. So what about the monkey? Do we think that there are any kinds of similar kinds of signals in the primate medial temporal lobe? Well, we don't yet, although we do have plans for this, we don't yet have our monkeys moving around in open arenas to try to look at comparable signals. But in our experiments, you remember we had the monkeys moving their eyes to explore scenes. And this was work with a graduate student, Nathan Killian, at Emory University. We had the idea that maybe as the monkeys are exploring these scenes, they're doing something similar to rats as they're moving around and exploring their environments. So as primates, we can get a lot of information about our world from moving our eyes without having to actually physically visit a place. And so we wondered whether we might see spatial representations that reflected this kind of visual exploration. And I always like to pause here and say a minute about being honest about our methods. This is an old Twitter meme that was quite popular for a few days. This is overly honest methods, where people sort of went on Twitter and confessed about their methods. This is one that I really like too. We don't know how the results were obtained. The postdoc who did all the work has since left to start a bakery. There's another good one that said, we recorded in the hippocampus because it was big and we didn't know where else to look. But if I were going to do our overly honest methods, I would say that we didn't set out to look for these kind of spatial representations. As you know, as I mentioned, we were really studying memory and we used the eye movements as an index. And it's kind of a tricky signal when you're trying to record from neural activity and you're allowing the monkeys to move their eyes. Most experiments with primates really have the monkeys fixating and not moving their eyes. because moving the eye movements sort of caused some confounds in analyzing the data, which we thought we were just going to have to make our way through because we needed that eye movement for the behavior. But with Nathan Killian, as I mentioned, this graduate student, we really found that these eye movements allowed us to explore a lot more about our signal. As I mentioned earlier, we got to see this kind of rhythmic activity that was related to the eye movements. And in this experiment, he was able to look for these spatial representations. So just to give you a sense of the setup, in this experiment, Nathan was recording from a probe that had multiple contacts along the shaft, so he could record from all layers of the entorhinal cortex simultaneously. And he was recording in the entorhinal cortex while the monkey performed the same free viewing task that I described to you just a moment ago. And surprisingly to us, he found evidence for grid-like activity in entorhinal cells. So this is an example of a single entorhinal neuron that he was recording. The gray trace here represents the monkey's scan path as he's viewing 200 complex images in a given recording session. And these scan paths are just superimposed on top of each other. So we're just collapsing all the data together. And then the red dots just show where the monkey was looking when this single neuron fired an action potential. And this is the heat map of the activity. And then in the autocorrelation, you can see the 60-degree symmetry, which gives you evidence for how clear the signal is and how much you could draw these equilateral triangles between the firing fields. And so this is an example of the grid-like activity that Nathan was able to find in the monkey entorhinal cortex. But one really interesting thing about grid cells in the rodent is that they change their scale and spacing as you move through the structure from dorsal to ventral. So in the dorsal part of the entorhinal cortex, you can find firing fields that are small and tightly packed, and then they get larger and move farther away as you move to more ventral recording locations. And having these different scales has also been used in a lot of the models of how these signals might be important for navigation. In the monkey, the comparable orientation, given the difference in anatomy, would be moving from lateral to medial based on the rhinal sulcus. And so we looked at Nathan's recordings that were close to the rhinal sulcus, and we found that the fields were pretty small and tightly packed. And then as he moved away from the rhinal sulcus, we found that the fields got larger and tended to move farther away. So again, comparable to what had been described in the rodent. And in subsequent work, we also were able to identify border cells in the entorhinal cortex. So these are cells that fired whenever the monkey looked at one image of the screen. And also what we called saccade direction cells, which we think are comparable to head direction cells. So these are cells that were selective for a particular trajectory of the eye movement. And then just recently, with a graduate student, we went back and recorded from the from the hippocampus to see if we could find similar kinds of place cells in this free viewing task. And so this is work by Seth Koenig, who's a recent graduate from our neuroscience program here at UW. And in his work, he recorded from the hippocampus while monkeys performed this free viewing task. And here are just two example neurons where he found representation for a specific region of the screen. So here, this neuron tended to fire whenever the monkey looked at the upper right corner. And this neuron tended to fire when the monkey looked at the lower left corner. And this is just showing in red the response whenever the monkey's eye went into that field compared to in blue when it was out of the field. These are raster plots, so each little tick mark represents an action potential. In red, again, for in the firing field, and blue out of the firing field. So you can see the reliability and the selectivity of this response for a particular location. And just one field compared to the entorhinal cortex, where we found the multiple grid-like fields. So taken together now, I think we've identified all of the components of the system that had been previously described in rodents. So at least we know that across species, you have the same kind of building blocks. So how do we then think about bridging the gap between these two views of the system? Well, there was a review article that was published a few years ago, and several have sort of thought about this idea that maybe the way we think about the same structure being involved in these two different cognitive functions is that the neural mechanisms that support memory evolved from the mechanisms that support navigation. So something like episodic memory, which is memory for experiences that we've had, our autobiographical memories, maybe that kind of memory, or those processes that support that kind of memory, evolved from processes that supported egocentric navigation. So navigation that required path integration, self-reference navigation. And here's an example. This is the review that I was mentioning by Yuri Buzaki and Edvard Moser, where they sort of made this, or suggested this idea about the evolution of these processes. So the egocentric self-reference navigation may have evolved to support episodic memory, and more of a map-based allocentric navigation may have evolved to support semantic memory, so memory about facts. Well, if that's true, if you think about that these mechanisms may, that the spatial mechanisms may have evolved to support the memory mechanisms, then that suggests that you should be able to see these spatial representations when you're doing some kind of mental travel, even without any movement, and even in our case, without eye movements. And so in order to address that issue, or to see if we could at least find evidence for that first principle, Nicholas Wilming was a visiting graduate student in the lab, and he decided to train monkeys to fixate a central cross, and then they had to pay attention to a stimulus that was moving around in the periphery. And we knew that they were paying attention to it because every now and then there was a subtle luminance change in this stimulus, and the monkey had to indicate that he had seen that change by releasing a bar in his chair. And so if the monkey was correct in releasing the bar on time, we knew that attention was on this stimulus. And we could control the path of the stimulus in the periphery while the monkey was continuing to fixate, so not moving his eyes and not moving around anywhere in the environment. And Nicholas recorded from the entorhinal cortex while the monkey was performing this task, and surprisingly found evidence for grid-like activity, even in this case of just covert attention without any movement. So it suggests that these signals can be used or can be identified in the case of exploration or navigation in some ways without any kind of physical movement. Now, thinking about the grid-like activity and how it's conserved across evolution, so to date, it's been described in several species. So it was first identified in rodents, the work that I described by the Mosers. A few years later, the same group discovered these grid-like responses in the mouse. And then work by Nakam Ulanovsky's lab discovered grid-like activity in the flying bat. Our work was in the non-human primate, in the rhesus monkey. And then there's work by Josh Jacobs and Mike Kahana, which have identified grid-like activity in human epilepsy patients. And so, again, this activity has been described across species, but we're still at a stage in the field where we don't understand exactly what these signals are useful for. The predominant theory, I would say, has been that they're serving navigation, but it doesn't really fit with what we find from human patients who have damage to these areas, where navigation is not the predominant issue. It's more of a memory concern. So that's where we get to the third part, where I'm going to talk about something called the cognitive map. And another way that people have thought about these spatial and mnemonic signals, and a way to understand how the structure could be important for both, is to think about how these signals might reflect kind of a full map of cognition, not just space, but also other aspects of cognition that could be important for memory formation. So I'll explain that again in just a few minutes, but let me start with all the signals that we had shown so far for spatial representation had been identified in a 2D space where the monkeys are just moving their eyes and looking at two-dimensional stimuli, which is different from what has been the sort of experiments that have been done in rodents, where they're really moving around in physical space. And it's still technically challenging to do experiments where we let monkeys run around while we're recording. And so in the meantime, in order to try to get closer to the 3D space, we've trained monkeys to perform tasks in virtual reality, where we train them to use a joystick to navigate different kinds of three-dimensional arenas. I'll show you an example of one of our early experiments here. So this is our monkey Giuseppe, who's using a joystick to navigate this virtual environment. And his job is to move around the environment and try to find the bananas, and then move his joystick to collide with the banana. And every time you hear that beeping sound, the monkey is actually getting a food reward delivered through a tube. And the circle on the screen here represents the monkey's eye movement as he's exploring and trying to look for bananas. And so with this, we found lots of interesting behaviors. So you may have noticed the monkey was looking around or moving around, and then as soon as he identified a cluster of bananas, that's when he decided to go forward to maximize his movements there. We also found evidence that the monkeys could remember the location of the next closest banana. So you'll see even when it was off the screen, he knew which way to go next to find the closest banana. So lots of interesting behaviors. And at the same time, we've moved into new recording techniques where we're trying to increase the number of individual neurons we can record simultaneously, and also trying to record across the full length of the hippocampus, as well as the underlying entorhinal cortex, again, simultaneously, so that we can try to understand if there are differences in the signals that we see across the structure and then between the hippocampus and the entorhinal cortex. So this is a microdrive that was developed by Charlie Gray and his group, and we helped validate this for recording deep in the brain. And so this microdrive has 124 individual wires that are each independently movable, so that we can lower them down into the hippocampus and then adjust their depths to try to get closer to individual units to be able to isolate single cells. And this is just an x-ray from our first monkey showing that we've got about half of the electrodes in the back here lowered down to the hippocampus, so we can see that they're moving straight down to our target for the most part. And we've also done a lot of work in gaming, in developing new games for the monkeys in a Unity environment now. So we're using environments that have more rich visual stimuli and also are memory dependent. And I'm going to tell you about one of those today, where the monkeys were trained to perform an alternating, it's called an alternating Y maze, or spatial delayed alternation task, where the monkey's job is to move down the center arm and then on alternating trials, move to the left or right in order to get a reward. So I'll show you a video of one of the monkeys performing this spatial delayed alternation task. Okay. So this is our monkey Gromit. He's moving down the center arm. And then he took a right turn, and that was correct for this trial. So he gets to get a reward when he finds the banana hidden at the end of the hut. And that popping sound you're hearing is a single hippocampal neuron. And so hopefully you're hearing it. So you could hear that the neuron was firing at the choice point right at the end of the center arm. And then it fired again when the monkey got to the left side. We'll do a couple more trials. So it was pretty quiet there on the right side. It fired at that choice point and then kind of didn't fire too much more on the rightward turn. And there you could hear it firing a lot again, right at there, at that leftward, at the end of the leftward arm. And so, and that's an example of what we would call a place cell, or what looks like a place cell in the monkey. And we found many examples of these with our recording. So here's another one. This neuron tended to fire just after the monkey made the rightward turn. These are raster plots showing the left trials and the right trials. So these black tick marks indicate action potential. So this neuron fired a lot right after the center point on the rightward trials. The red tick marks here indicate incorrect trials, where the monkey turned the wrong direction. It's interesting to us for this neuron that it continued to fire for the incorrect turns as long as the monkey was going right. But interestingly, when the monkey turned left but was incorrect, this neuron also fired a little bit, making us think that, that perhaps this neuron was actually coding for what the monkey thought was a rightward turn. We also have been able with virtual reality to do experiments looking at what's called remapping. And so one prominent feature of hippocampal place cells is that they tend to show a different kind of response when you move the animal into a different environment. And so in our case, we can, within a given recording session, we can move the monkey into a different environment by just changing the visual appearance of the virtual environment. And so what we typically do is we record in one environment, then move the monkey into a new environment, and then go back to that initial environment. And here are two example cells that show strong place fields in one environment. In this case, you can see that it's reliable across multiple presentations of that environment, but has a very different response when the monkey is doing exactly the same task, but just in a visually different space. So similar again to the place cells that have been described in the rodent hippocampus. So now if we go to thinking again about how we bridge the gap, so now we have all these spatial representations that have been identified in the monkey, even in now 3D virtual space. So do we think that these responses are important for navigation? Well, no, I mean, my bias is still to always go back to the mnemonic representation and try to think about how these signals might be important for memory, because again, from my experience with patients, that's the primary issue when you have damage to this area. And so, as I mentioned, there's another idea that maybe the hippocampal formation serves as what's called a cognitive map. And what it does is it organizes our experiences and guides behavior across all domains of cognition. So it just turns out that space happens to be an easy way to align activity, but that you might get a similar kind of mapping if you could just align all the content of cognition. That what the hippocampus and the infrontal cortex are doing is just sort of providing a structure of all that kind of experience and all the, sort of a map of knowledge or a map of cognition. And so if that's true, then we should be able to come up with experiments where we could train the monkeys in some other kind of environment where we're controlling the content of cognition that's not spatial. And this was actually the same idea that David Tank had, and he published this work a few years ago in 2017. Dmitry Aronov is the first author. And in this experiment, they trained rats to push a lever. And whenever they pushed the lever down, there was an auditory sweep that occurred, so a tone sweep that changed in its frequency while the rat continued to hold the lever. And the rat's job was to release the lever whenever the frequency got into this target zone. And recording from the hippocampus, they found that there were cells that were selective for particular frequencies, kind of mapping this auditory space in very similar to the way that these hippocampal place cells would map real space as the rats were moving around. So indicating that they could find evidence for this kind of mapping of content that's outside of space, but just another aspect of cognition. In our experiments, this is work done now with Yoni Browning and John Rookman in the lab. We decided to tackle color cognition or color space. And so we trained our monkeys to use the joystick to navigate this world that we defined entirely by color. And so just to give you a sense of how this task works, the monkey's job is to move the joystick through this world of color that he's learned, and release the joystick when he gets to the color that matches the sample, the one that's around on the outside. So in this case, show another trial here. The monkey is looking for the green stimulus, and the monkey knows which way to move the joystick to get to that green stimulus and then release as soon as he gets to that point. In new experiments, we've been able to show that we can train monkeys on new sort of configurations of the color, so new color maps each day. And we're starting to record now in the hippocampus and entorhinal cortex as monkeys perform this task. So along with representations of kind of space or the color, the stimuli that the monkey is seeing, we also see lots of other kinds of responses in these VR tasks. Here's an example of a few other ones. So we see responses for the reward. So when the monkey gets to the right place and is approaching the banana, which is time 0 here, and we see reward responses that are selective for which side of the arm or which side of the maze the reward is on. So this neuron fires more for approach to reward from the left than from the right. And we also find responses that in the rat have been called time cells. So these are cells that respond at a particular time within the interval between trials. So that was that period where the screen goes black and the monkey just has to wait a few seconds for the next trial to start. And this neuron shows the selectivity for firing just within a few hundred milliseconds after that period starts. And we find neurons that tile this entire time of the delay interval. So responses really that map kind of any aspect of the experience that the monkey is having, any aspect of the task. And this leads to kind of the final idea of how we might think about bridging the gap between these two views. And here we're strongly motivated by another review by Yuri Buzaki. This is Yuri Buzaki and David Tingley, published in 2018. And showing here, each color represents a different place cell that was recorded in the hippocampus. Here the rows represent different trials as the rat is just moving down a linear track. And so what you can see is that if you look at each different color, you could tell that these are place cells. So these neurons tend to fire in different places as the rat is moving along the linear track. You could also just align the activity based on the time that the rat has, the time that's elapsed since the rat started moving down the track. So you can see that you can get kind of a representation of time as well. But really what's most striking is when you instead take the activity and align it relative to this more internal signal of a theta cycle. And here you can see that these neurons, or the firing of the neurons, really line up beautifully across these different theta cycles. And you can see this progression of a sequence so that these neurons are kind of firing in sequence and sort of going back and then continuing forward on each of the subsequent theta cycles. Now, we don't yet have a great sense in the monkeys of what this kind of internal timing signal might be, the way that these responses might be structured to look for sequences. But this is exactly where we're going in our ongoing and future studies. I'll just give you one hint of this that we found that seems like it might relate to this. And I'll go back to the work by Seth Koenig, where he was recording from the hippocampus and identified these place cells that reflected where the monkey was looking. When we started looking in detail at all of the different hippocampal place cells that we found, we noticed that their latencies could be different. So here I'm showing 4 different cells. Again, the red trace shows the response of the neuron when the monkey moved his eye into the firing field, the location that this neuron was selected for. And what we find is that some neurons responded just as soon as the monkey's eye went into that field. and other neurons had a much delayed response, so very different latency. And if we look across the population, so now each row represents a different cell, a different neuron. The dotted line here is the time of the fixation, so fixating into the view field. And the color represents the intensity of the firing for each of these neurons. And we've just sorted them by the latency of their maximum firing rate. And what you find is that we see neurons that have latencies that completely tile the time of the fixation. So within that 200 milliseconds where the eye is not moving, and presumably that's when all the incoming information is being processed through the system and getting into the hippocampus, we see responses that are showing a really tightly timed response relative to that fixation start. And across the population, they give us evidence that this might represent a kind of sequential firing of this hippocampal population. Now, one caveat is that these data were recorded using our old techniques where we were recording sort of 4 or 5 neurons at a time. So we can't really clearly say that these represent neural assemblies or sequences, but this is something that we're going to be looking at now in with our new system where we can record from tens to hundreds of neurons simultaneously. And so this really leads to kind of the going idea in my lab and some others in the field that the hippocampal activity really reflects the experience of the organism, not objective reality. And it's really kind of wrong-headed in some way to try to take aspects of the world like time and space and even the stimuli that we're showing and try to align the neural activity to the features defined by the experimenter. Instead, we're going to be better able to understand the organizing principle of the hippocampus if we can find that internal rhythm and look for those internal sequences, those internally generated sequences. And so that's kind of where we're going in our, as I mentioned, in our current and future experiments. So I'll just conclude there, just to summarize a little bit of what I showed you today. So the first piece was that visual exploration, moving our eyes for monkeys and humans, is a way of actively sampling the world. And we see a reflection of that behavior in the activity in the hippocampus with this sort of alignment to a low frequency theta band oscillation. In the monkey, we've also been able to identify spatial representations relative to these eye movements. And also the last part I told you in the temporal representation. So these responses that have a fixed latency relative to the time of the eye movement. And then in our future studies, including these large-scale recordings throughout the hippocampus and these novel tasks in virtual reality, we hope have the potential to increase our understanding of the nature of what we mean by a cognitive map, and more importantly, I think the processes in the hippocampus and internal cortex that are guiding memory formation and trying to get a better understanding of the structure of those representations. I just want to end with one final video, just showing you how much we can really tap into in terms of the cognition of the animal by using these virtual reality tasks. We had a monkey that was fully trained for many, many months on doing that kind of open field navigation, looking for bananas in a pretty sparse kind of environment. And one of our technicians was working on her ability to create new environments. And so she made this really rich, beautiful environment. And we decided to test it out with the monkey. And so what I'm going to show you is the first day that the monkey was put in this new environment. So you're going to see the monkey moving around. This white dot is the monkey's eye movement. And the monkey was food motivated. So he hadn't been fed since the day before. He knows exactly what to do to move around to get bananas. And then, at the risk of anthropomorphizing, it's as if you suddenly can see that the monkey realized that he was in a different environment and just sort of stopped. He's not moving at that point. He wasn't moving forward. He was just looking around. And now he starts to approach kind of the new stimulus, completely forgetting about the bananas at this point, or they're just not as motivating, even though, again, he hasn't been fed since the day before. But he's just, and I always get sad when I see them bump against the wall there. You really want to root for the monkey to be able to get out and explore the rest of that environment. But the monkey did this for over 2 hours, just kept moving around and exploring this new rich environment. And so one thing that we're really interested in, every now and then he'll go back and get a banana, especially if it's kind of right along his path. But we're really interested in taking advantage of these new techniques for being able to record from large populations of neurons to be able to look at the activity that might reflect this kind of novel learning where you don't have trial structure, but you just have this, this sort of more naturalistic behavior. Okay, so I'll end there. I want to thank all the people in the lab who contributed to this work. I mentioned their names along the way, just to point out a few. John Ruckman and Yoni Browning are really spearheading the new work with virtual reality. And Megan Jutras, see, where did she go? Right here in the middle. Megan Jutras is my longtime lab manager who really helps keep everything going. I thank our funding sources, and I'm happy to take questions.